Not so much. I'll use the microphone. Okay. Okay, so what are we going to talk about today? The stem cell compartment, essentially. Now, and a few things about how the stem cell compartment is regulated using the model system of the mammary gland and therefore of the stem cell compartment in the mammary gland. And then we'll try to see how things can go wrong in tumors in the stem cell compartment, in according to an hypothesis which has become very popular in the last few years, which is the hypothesis of the stem cell origin of cancers, so that the cancer is actually a disease of the stem cells in a particular compartment. Now, you all have an idea about what cells dream of. When a cell is sitting there, somewhere, in a culture plate or in a body, anywhere, what does it dream of? The only dream of a cell is, of course, to become two cells. That's what drives evolution. Selection. The more progeny you make, the better off you are. So the dream of a cell is to become two cells. But when we now go and try to extrapolate this very simple and basic concept to how complexity is built out of cell replication, then we run into some unexpected findings. So, what is the relationship between proliferation and differentiation in an organ? When you build an organ, any organ, I will talk mostly about breast today, but you can imagine the same thing for any other organ of your choice. Well, the entire mechanism is a little bit more complicated than cells just dividing and then deciding in a mysterious way to assume a differentiated fate. Everything starts with a very limited number of cells, which is present in every organ, and here we are talking about adult organs, so we are not concerned about embryogenesis, we will not concern ourselves with the embryonal stem cell. Everything starts with this limited number of cells, which we call the stem cells. What is the peculiarity of stem cells? The absolute peculiarity of stem cells is that they are capable of performing what we call an asymmetric division. What does it mean, asymmetric? It means that the two progeny of the division of the stem cell are not equal, they are different. Now, how this is achieved, it's something that we will try, at least in part, to explore in the rest of this lecture. It is important for you to understand that at this point, the definition of asymmetry that I'm giving you is a functional definition. It's not yet a molecular definition. One of the things that we'll have to try to do as we move on with the lecture is to try to give some molecular support to this functional concept. But the concept is pretty simple. The two daughters are different. Now, it's not immediately evident how can you take the same genetic material, divide it in two daughters in a manner which is identical, because the genetic material of these two progeny is of course identical, and still give rise to different progenies. We'll try to see how this happens. But at the functional level, the consequence of this asymmetric division is that one of the two daughters retains the stem cell fate, while the other daughter becomes what we call a progenitor. Now, in functional terms, what is the difference between this daughter and this daughter? Well, functionally, the first important difference is that this daughter withdraws immediately in quiescence. This is a characteristic of stem cells. Stem cells replicate very rarely. the progenitor, on the other side, immediately starts proliferating. And it proliferates with a pattern of division which is largely symmetric. Means that the daughters of the progenitors are all equal to the mother. So there are two major differences. One is that these cells will retain all the traits of a stem cell, which means quiescence, and eventually, when this cell will re-enter the cycle, sometime after weeks or months or years, it will divide again asymmetrically. The progenitor, on the other hand, kind of explodes in its ability to proliferate, and it goes through a number of rounds of division which are different from organ to organ, and generates a large compartment. This is called the transit amplifying compartment, amplifying because the number of cells amplifies, and transient because it does not last forever. Because eventually what happens is that all the daughters, all the progeny of this initial progenitor will differentiate. Will differentiate acquiring the characteristic of the mature cell of a particular, of the particular organ in which that division is happening. So at the end of the day, when you look at an organ is essentially made of all the daughter cells stemming from the progenitor, because this is where the cells come from in the bulk of the organ. But since they are all differentiated, they cannot repopulate the organ, and to repopulate the organ, you have to start again from the stem cell, from the very few stem cells that are somewhere. This division is a tricky one. Things not always go well in this division. More or less enough of the cases, as we calculate right now, things can go wrong. And these cells can either undergo apoptosis or senescence or something like that. So if we put together all this evidence and the concept that we just enunciated, which is stem cells divide infrequently and do not expand in number, because from one stem cell you can only get one stem cell, then from the experimental point of view, if you were able to count stem cells over time, you would expect a behavior which is somehow between these two curves. The theoretical behavior, if everything goes fine, is that the curve should stay flat. From one stem cell, you can only get one stem cell. And the measure of time could be anything. Could be real time if you are really able to count stem cell in an organ. I will show you that this is possible. Or in an in vitro equivalent, you can passage stem cell, and I will have to show you how you can actually passage and count stem cells. And so you can count them over passages in vitro, which is is an experimental surrogate for time. So this would be the theoretical curve, but most likely you will observe a curve like this, because at every division you tend to lose a number of stem cells. This is what we call the self-extinguishing behavior. This is what we call a mortal behavior of the stem cell. And current thinking holds that basically in every adult organ and tissue, stem cells are not immortal, but they have a limited lifespan. And that's the reason why most people believe that we age. We age simply because we run out of stem cells, and so we cannot repopulate our organ efficiently, so we end up losing organ function. So, in a way, and to use a popularization of the concept, really tissues are organized as beehives. So a tissue is made of a vast multitude of workers, which are, however, for all practical purposes, sterile. And so the beehive can only be repopulated by a queen, which in our case is the stem cell, or the few stem cells present in a tissue. This has an interesting implication from the cancer point of view, and we'll try to get to that. Because if you want to exterminate a beehive, you can kill as many bees as you want. The beehive will reform very shortly. While if you want to really kill the beehive, you have to kill the queen. Now, when we will try to translate this to cancer, you will see that this leads us to a number of paradoxes which might actually have affected in a profound way our interpretation of drug efficacy. Let me open a small parenthesis and then we'll try to get to some more cell biological concepts. But how do we test the efficacy of a drug in the clinics? We give the drug to the patient and we look at the shrinking of the tumor mass. Now, the shrinking of the tumor mass in a beehive corresponds to the massive killing of the working bees. Might not have anything to do with the actual ability to sterilize the beehive. On the other hand, imagine the theoretical possibility that we develop a drug, and I will try to show you that at least as a proof of principle this is feasible, that we develop a drug that can selectively kill stem cells. You give this drug to the patient, and what is the result? You see no effect. For the period of time in which you observe the patient, which is normally a short period of time, unfortunately, you know that these studies are done, especially for new drugs, are done on patients which are terminal. So basically, there is really no hope of curing them because they're already vastly metastatic and stuff like that. We will not observe a major improvement if we measure tumor mass. And so we would throw away the drug as non-efficacious, while we would retain drugs which are toxic and killers for a population which is likely not the one that we should really be interested in. But we try to go again on these concepts. So let us ask ourselves now, well, this is what happens in normal tissue. But what happens in cancer? The popular misconception, and you don't have to consider that as students you have a misconception, because I'm afraid that a lot of colleagues of mine still will have this misconception. It's so enrooted in the way we have been brought up, is that cancer is a bag of cells. Cancer is a bag of cells that have lost control, they simply sit there, happily proliferate, don't give a damn about the health of the host. The only reason for the short term is in evolutionary term, they get their advantage and they live out of that. Well, not true. Cancers are actually organs. As I will try to show to you, this should not surprise you if you think that cancers originate from stem cells. So if cancer originates from stem cells, what are they? They are simply organs that are formed by a cell, a cancer stem cell, which is programmed to give rise to an organ. But simply does it in an aberrant way because some of the pathways present in dead cells are not perfectly functional. So in a way, the fact that cancers are organs, it's something that we can already understand by simply looking morphologically at cancers. Some cancers are very well differentiated. This is from a mammary gland. This is an area of cancer. You see how well differentiated this area is. I mean, this looks almost like a normal gland. There are alveoli, the luminal cells are in a single layer. You know, it's not exactly a normal gland, but it's very close to it. This is what we call the well-differentiated cancer. Then there are cancers which are totally disorganized. We call them poorly differentiated cancer. The amazing thing is that frequently, area of good differentiation and bad differentiation are present in the same cancer. I will not dwell much into this, but I want to use this image to give you some visual of the fact that actually cancers are simply aberrant patterns of morphogenesis, but you should consider them like organs. They do exactly the same thing that organs do. So, if cancers are organs, deviate it as you want, but organs, how do they originate? Well, according to this idea, they should originate from a stem cell. But what is the difference between a cancer stem cell and a normal stem cell? Well, here I will get a little bit ahead of myself and present you some summary which is also in part due to the work that Salvatore and I have been doing, and that I will try to present later, but since it's also due to the work of a lot of other labs, it's fair to present it in an introduction. We believe that among a lot of other differences, don't get me wrong, whenever people give a seminar, they will give you their way of looking at the problem, but there are several other ways, of course, of looking at the problem. One of the differences, and what we believe is an important difference, is that cancer stem cells have acquired a novel property with respect to normal stem cells, and it's the fact that they can divide symmetrically. Now, in the stem cell compartment, a symmetric division entails that from one stem cell, you give rise to two stem cells. The first consequence of this is clear. Cancer stem cells can actually expand in number, because from one cell, you can make two cells. Is it an obligatory pattern? No, it's not. If the stem cell would always duplicate symmetrically, then cancer would really be a bag of cells. They would all be identical to themselves. Now, in cancer, what happens is that the stem cells has acquired the dual capability of either dividing symmetrically, giving rise to two stem cells, or dividing asymmetrically, remembering what it's supposed to do as a well-behaved stem cell. And so in the asymmetric division, it will still divide, giving rise to a stem cell, and then to a progenitor that does the same voodoo that I just explained to you in the normal tissue. So at the end of the day, the cancer tissue is going to be made mostly of progenies coming out of the progenitor, which differentiates, frequently differentiates in an aberrant way, because the molecular program in these cells, of course, are subverted, but will also have an increased number of stem cells. And actually, what Salvatore and I demonstrated a few years back is that the aggressiveness of cancer correlates with the number of stem cells. So essentially, the higher the propensity of a particular cancer to skip rounds of asymmetric division in favor of rounds of symmetric division, the higher the stem cell number, the higher the aggressivity of that particular type of cancer. And so now we can rationalize in a similar kinetic curves as the one that I showed you for normal cell, what should be the behavior of cancer stem cells. Cancer stem cells, we expect to see them increasing in number over time, be time something that you assess directly in vivo, or something that you assess in vitro with passages in culture. So the question now is, how do we reduce a stem cell model to something that we can handle and manipulate in vitro? In science, it always boils down to that. If you cannot transfer something into a system which is amenable of being studied in vitro, or at least in an ex vivo condition in which you can continuously go from in vivo to in vitro, then you are bound to the impossibility of perturbing the system and therefore to achieve conclusions. Well, one interesting problem which I will try to explain to you here is how do you count stem cells? In the theoretical curves that I showed you before, there was somehow the assumption underlying that I was able to count stem cells. But now, how do I really count stem cells? Think about it. Imagine that I have a purified stem cell, I put it in culture, the cell starts to proliferate. All the progeny is going to be progenitors. My stem cell is going to be immediately diluted among the progenitors. So how do I keep track of the stem cell? How do I follow the stem cell? How can I count them? Well, there are tricks, of course, that you can use. This trick here is used in the breast stem cell compartment. It's used also in other compartments. It's based on a peculiar characteristic of the stem cell to be able to withstand anoikis. Now, what is anoikis? normally cells to grow have to adhere to a substrate. Be it an in vitro substrate, like the one we provide the cells, plastic, fiber-active, glass, you name it according to your experimental needs, or an in vivo substrate, like a basal lamina, or things like that. When cells are detached from the substrate, with few exceptions, like blood cells, for example, they are incapable of surviving, or at least incapable of proliferating. Stem cells have the ability, and this process is called anoikis. It's lack of housing. Oikos is the house in Greek. Now, in modern Greek, actually, because it comes from Turkish, the ability to withstand anoikis now, it's something that we can use. If you have a gland, a mammary gland, and you dissociate it, and you have a rare stem cell in the midst of progenitors and differentiated cells, and you plate these cells in suspension, in a condition in which the cells do not attach, only the stem cell will give rise to a clonal expansion, to a spheroid, which we call a mammosphere. Now, in a mammosphere, in general, you have only one stem cell, for the reasons that I explained to you, because all the rest is going to be made of progenitors or of cells that start to undertake their differentiative way. But the interesting property is that every spheroid, and that's the way they will look in real life, is the result of a single stem cell. So now you have a way of counting stem cells, because simply you dissociate the organ, you make the spheroids, you count them. You know how many spheroids, how many stem cells were present in the original gland. Then you take all the spheroids from the first generation, you dissociate them, you plate them again, you let the spheroid emerge, you count them. And you can go on and on and on. So by passaging and not counting cells, by counting spheroids, wait. By counting spheroids, you can actually follow stem cells. And so let's get to some experiment, actual experiment, to prove you that what I'm telling you is true. This is an experiment that we have done a few years ago together with the group of Pier Giuseppe Pellicci, is comparing the in vitro ability of mammosphere to propagate over passages, like I just told you, in two different conditions. One is a wild-type condition, so these are mammary glands from the mouse, dissociated and put in the mammosphere assay. And these are mammary glands from a mouse which has been engineered by ectopic expression of a very potent oncogene called ERB2. This is a transgenic mouse. And these mice have high propensity to form tumors. So this is actually a tumoral gland. Well, you see that basically you plate the stem cells from the first passage, and they will give rise to a few spheroids, but they are less than what you plated. At this point, you replate, you concentrate these, you replate the same number of cells, and go on and on and on. So you see that at every passage, fewer mammospheres than before will fall. They will still fall. But if you look at the cumulative number of mammosphere over time, this is exactly the kind of curve that you get. So this doesn't mean that these cells are not proliferating. They are actually proliferating, but at every round, they are proliferating less than before. And so cumulatively, you get fewer mammospheres. You do the same kind of experiment on the ERB2 mammosphere, and I mean, I don't need to go into details. You see that every time they expand, and so the cumulative number will be a logarithmic growing curves, and will define what we call an immortal behavior of these stem cells. So this is one type of experiment, the mammosphere experiment, that I will use frequently in the rest of the talk when we get to the more experimental part. Basically, the take-home message here is that when you see a curve going down, that means that the behavior is self-limiting and that the cell is behaving like a normal stem cell. When you see the curve going up, it means that the cell is expanding, that the behavior is immortal, and that that stem cell is behaving more like a cancer stem cell. There is another technical problem than one might want to challenge that Salvatore actually challenged a few years back, which is, again, you take a stem cell, you make it proliferate into a mammosphere, and what do you get? You get 400 progenitor in one stem cell. So if you really want to study the stem cell, how do you do it? How do you isolate and purify stem cells? People have been devising all kinds of methodology, mostly based on antibodies for antigens supposedly specific for stem cells to do immunoaffinity-based purification of stem cells by FACS sorting. Bottom line is that these things don't work very well. Yeah, it's true, they enrich the population. So they can enrich your population, to 5-6% of stem cell. It's a good result. In a mammosphere, you have 0.2, 0.3% of stem cells, 1 out of 300. By doing immunoaffinity-based purification, you can increase that number tenfold, perhaps 20-fold. But it's still not good to do a lot of the things that we want to do. It's good to do biology, for example. You can transplant the cells and really prove that you increase the number of stem cells in there. But if you want to do biochemistry, if you want to do molecular biology, you need pure populations. So what Salvatore did a few years back was to adapt a methodology that had been actually widely used in the hematological field, which is very simple, and it works because it's simple. He takes a dye of the PKH family. You will see that these dyes come in different flavors. They can be either red or green. The important thing to remember is that they are fluorescent. This is something that we will use later in the experiment. Takes a dye, which in this case is PKH26, which is red, a dissociate mammary gland, and it stains everything. Now, this is a lipophilic dye. What it will do, it will intercalate in the membranes and stain the membrane. And will stay associated with the cell because the lipid trap the dye, and even if when the membrane are internalized, the dye will get in the cell in association to intracellular membrane, but keeps on staying associated with the cell for several generations. The other thing is that once you have stained the cell and removed the dye, there is no more supply of the dye. So what you got is what you will have for the rest of your life. Now, this population, which is more or less homogeneously stained with the dye, is now put in suspension culture. Most of the cells which are not stem cell will die because of anoikis, as I told you. The stem cell will form a mammosphere. But now, in this mammosphere, something interesting happens, because, let me go back a couple of slides. Yeah. So the first stem cell here will be fully stained with the dye. We'll have 100% of the dye. At the first division, this stem cell will get 50% of the dye, and the progenitor will get 50% of the dye. But then after that, the progenitor divides at least tenfold. And so progressively, the dye gets diluted, so that every cell gets less and less dye over generation. And at the end of the process, all the progeny is dull. It's not bright anymore. But the stem cell is very bright, because it has 50% of the dye. So at the end of the day, you get a mammosphere which looks like this. All the cells are dull, they are progenitors, and you see embedded here in the thickness of the mammosphere, there is one bright red cell on a good day. On a bad day, sometimes you get two per mammosphere. In the average, you get one cell per mammosphere. So the interesting thing now is that you can harvest all your mammospheres, dissociate them, and FACS sort. And so you can harvest a PKH positive and a PKH negative population. And we proved, and I will cut the story for you because it's published, so perhaps you can discuss it later with Salvatore, we proved that only the PKH positive population is actually made of stem cells. They have all the properties of the stem cell. You can use them, you can reform the mammary gland in the mouse by starting with a single PKH positive cell, while the PKH negative do not have the properties of stem cell. The important thing is that, again, on a good day, this population can be up to 95% pure. So now it's a good population to try to do some biochemistry or some molecular biology. And we did some. I will be brief here because this is just to try to illustrate to you some of the applications that you can use once you have a reagent as powerful as this. We made expression profiles, for example, of stem cells, of mammary stem cells. So we did analysis of the PKH positive versus the PKH negative. We isolated a number of genes that you see very clearly distinguish PKH positive from PKH negative. So these are the first snapshots of the molecular differences between a stem cell in the mammary compartment and the progenitor in the mammary compartment. And what we proved is that actually these expression profiles can stratify cancers. What does this mean? This means that if you take a group of cancers which are otherwise homogeneous, that look indistinguishable from each other, and you divide these cancers into groups. One that, in terms of expression profile, so expressed messenger RNA, looks more like stem cell, and one that looks more like progenitors. And then once you have divided these two groups, you interrogate the two groups for how long these patients survive. You see that the group that has a PKH positive signature, which means the group that looks more like stem cell, has a much worse prognosis than the group that has the other signature, the non-stem cell, that lacks the stem cell signature. And this can be done on several cohorts and several collection of cases. Now, why is this? Why should a cancer look more like stem cell or less like stem cell? As I told you at the beginning, when I gave you the introduction, I was getting a little bit ahead of myself. And now this is the reason. The reason why is because actually some cancers contain a lot more stem cells than other cancers. So basically, by doing the profile, we are doing an average of every single cell. So we are saying this cancer in the average looks like this. But actually what we are doing, we are counting the number of stem cells in some cancers with respect to other cancers. And this you can prove in many other different ways. The most stringent ways of proving this concept is doing a limited transplantation experiment in tumorigenesis assay. So you take actual tumors and you implant into a recipient mouse serial dilution of cells. You implant 1,000 cells, 100 cells, 1 cell in a sufficient number of mice, and then you establish how many tumors develop, and then with the Poisson distribution, you can calculate the frequency of tumor-initiating cells in those cancers. Of course, by everything that I've told you, it should be clear now that only a cancer stem cell is a tumor-initiating cell. So if you implant a progenitor, when you go at the lowest dilution, the cancer will not develop, but when you implant a tumor stem cell, the cancer will develop. And you see that there is a huge difference in the frequency of stem cells between well-differentiated and poorly differentiated tumors. The poorly differentiated tumors having a much higher frequency of stem cell with respect to the well-differentiated tumors. So let's try to get more molecular now. What are we really talking about? If you ask a developmental biologist, there are weird people that spend their life with flies and worms and stuff like that have discovered a number of things that we would have never discovered in more complex organisms. So now we go there and we sack what they've done and we translate it into human beings, more or less. Human beings are not very different from worms, you know that. A C. elegans has how many genes? How many genes in a C. elegans? 17,600. How many genes in a human being? 22, 23, something like that. Not very different, isn't it? Most women will agree with that. Worms are not very different from men. But we can use the experience of developmental biologists to address important problems, even in mammalians. Now, if a developmental biologist will look at everything that I told you so far, will tell, well, you know, we've been analyzing this problem for the last 40 years. It's called self-fate determination. It means two daughters assume different fates. A developmental biologist will tell you, this is the default. If nothing happens in a cell, the cell gives rise to two equal progenitors. You need active mechanism to impose asymmetry and to impose different self-fate. So they will tell you, again, one very important lesson that tends to be forgotten in cancer research is that you have to study the normal mechanism to understand how mechanism gets subverted. And if you go directly from what happened in cancer, you're bound not to understand very much. Because this is the default, actually, in a simple paradigm of self-fate determination. Well, you see, is this helping us? Yes, this is helping us. This helps us a lot because by using the incredible power of genetics in this organism, in lower organisms, developmental biologists for the last 40 years have already accumulated enormous knowledge about self-fate determination. They know the determinants, they know the proteins. They have a first name and a last name to a number of proteins which are actually involved in the process. So these are the proteins that we should be looking at. The most interesting one, this is the most interesting one for two reasons. One is because it's the first one that has been discovered, so there is some importance. The second is because we have decided to work on it. But the reason why we have decided to work on it is because we ran into it, not because we actually decided that this was the most important one. It's a protein called NAM. In a second I will show you why this protein is called NAM. It's a very interesting story. But NAM has been known for many, many years to be a cell-autonomous, self-fate determinant. What does this mean? It means that when a cell that is ready to undergo an asymmetric division is ready to divide, NAM is captured by one of the two poles of the cell in association with the membrane. So when the cleavage plan occurs, one of the daughters inherits the majority of NAM, and the other daughter inherits nothing. So now this is very interesting, because now this is transposing the functional concept of asymmetry into an actual molecular concept of asymmetry. An asymmetric division is actually due to asymmetric partitioning of self-fate determinants within the cell. This is not the entire story. There are also non-cell autonomous determinants, like the niche, for example, the niche concept. I'm not going to go into it, but I will limit myself today to some determinants, in particular NAM, which are cell autonomous, which means are associated with the cell, or within the cell. Okay? How does NAM work? Well, for many years, people have been knowing from the genetic point of view that NAM is an antagonist of a membrane receptor called Notch. Notch is a surface receptor. It's actually made of two subunits. It interacts with ligands called Notch ligands, names like delta or serrate. These are part of the family of the Notch ligand. Upon the interaction, a series of proteolytical cleavage in Notch occur, and the intracellular domain of Notch is released. It moves to the nucleus and it acts as a transcriptional cofactor. Very simple biochemical mechanism. Well, it was known, it has been known for many, many years that NAM is an antagonist of Notch. But really, nobody quite understood how NAM could antagonize Notch, and how this antagonism was somehow involved in self-fate determination. And this is until we run into NAM, because of a completely different story. What the lab, still part of our lab, is doing is membrane trafficking and endocytosis. We discovered in a screening that NAM is an endocytic protein. Nobody had ever thought to self-fate determination in terms of endocytosis. But now it became very clear how NAM works. Let's go back to Drosophila. Now, in Drosophila, the NAM concept was established in a very simple system, which is made of one cell, which undergoes 2 duplication to give rise to 4 cells. And these 4 cells constitute the sensory organ of Drosophila. So the sensory organ of Drosophila is essentially a hair. Drosophila is full of hair, which is connected to a neuron with a couple of cells that hold it in place. Those are the 4 cells that originate from this initial cell. When the sensory organ morphogenesis goes awry, the Drosophila is numb. So the Drosophila doesn't feel, because the hair do not work, and so the Drosophila doesn't feel. That's the mutation that was discovered in this protein that was called NAM. How does it work? Well, it works essentially at the first duplication of the initial precursor of the sensory organ, in this cell called SOP, which means sensory organ precursor. When the cell divides, as I told you, NAM goes only to one of the poles, and so is inherited only by one of the 2 daughters. It's not inherited by the other one. On the surface of the SOP, there are Notch and ligands for Notch. There are also other proteins which are essential for Notch activity, but let's forget about it. So, in this daughter now, the presence of NAM, which is an endocytic protein, imposes the selective endocytosis of Notch. So Notch is cleared from the cell surface. Notch being cleared from the cell surface, this cell now will retain delta, because delta is present. It's not asymmetrically partition, but it has no Notch. On this other cell, Notch is present, because NAM is not there, and so there has not been any selective endocytosis of Notch. And now this cell can signal to this one, because it has the ligand, and this cell has the receptor. So now the 2 daughters become a signal-giving and a signal-receiving cell, and therefore they will develop different networking and different biological responses, and they will assume different things. So this cell is different from this now. They divide again, and they finally differentiate in the 4 cells that I told you. You remove NAM, all this thing goes banana, the 2 daughters are identical, the sensory organ doesn't work. Now, why is NAM very interesting? Well, for us, NAM became very interesting when we also ran into another observation. This is not only very interesting because it's an endocytic protein, we clarified how it works, but we discovered that the expression of NAM is very frequently lost in human tumors. In almost 30% of breast cancer, it's also true of lung cancer, but I will not dwell into it. In almost 1/3 of breast cancer, you see that NAM is completely gone. This is the staining for NAM in a normal gland. This is in breast cancer. These are actually normal glands trapped into the tumor, so you can actually appreciate the difference. So 1/3 are, as we call them, NAM deficient. At least 1/3 are NAM proficient, which means that NAM is not altered. And then there are some 1/3 more or less of intermediate cases. Now, Salvatore did a lot of work in his younger days, he also had younger days, to discover which is the mechanism underlying the loss of NAM expression in tumors. And what he found, very interestingly, that this is not a genetic alteration of NAM. It's a totally post-translational alteration, which involves hyperdegradation. So the bottom line, which is illustrated here, but I will be very quick, is that in these tumors, NAM is hyper-ubiquitinated, which means that there are enzymes that modify very heavily NAM with ubiquitination. You know, or you should know, that ubiquitination is the signal to target proteins to the proteasome for degradation. And that's the reason why NAM is expressed at very low level, not because it's not synthesized, but because it's very rapidly degraded. And as a matter of fact, if you treat the cells with an inhibitor of the proteasomal degradation, MG132, you see that you rescue completely the level of NAM in a way which is indistinguishable from what you see in normal cells. NAM is important in the natural history of the tumor. The loss of NAM correlates very precisely with prognosis. So if you have low NAM, you have a very high, much higher probability of having secondary events, essentially metastasis, with respect to tumors which are NAM proficient. So this is all in breast cancer. Now the story gets thicker when another brilliant guy in the lab, Ivan Kolaluka, ran into what at the time was a very surprising observation. So we have always interpreted NAM in terms of notch antagonism, and as a matter of fact it's true. I haven't shown you the data, but in these tumors, in the NAM low tumors, there are high levels of notch and high levels of notch activity, as one would expect from everything that we know. But he ran into another very interesting observation. NAM also regulates p53. This is a completely different story, completely unexpected. He ran into it for reasons which are too complicated to explain. But the consequence of his work, which also got published a few years back, so I'm not going to dwell much into this, are the following. P53 is normally regulated by an E3 ligase called Bdmv. In human, sometimes we call it HDM2, H standing for human. What is an E3 ligase? It's an enzyme that attaches ubiquitins to a substrate. So we go back to ubiquitination and degradation, like we've seen in NAM. When Bdmv2 interacts with p53, it ubiquitinates p53. Ubiquitination of p53 imposes degradation of p53. When NAM is present, it enters a tricomplex with Bdm2 and p53 and blocks the E3 ligase activity of Bdm2. So now this allows us to make a very simple equation. High NAM means high p53. Low NAM means low p53. Tumors with the high NAM, which are NAM proficient, are supposed to have high levels of p53. Tumors with low NAM, like breast tumors with low NAM, likely have low levels of p53. Well, this just shows, we'll go quick here because I'm actually running late. This simply shows you that what I told you is true, and that actually these tumors that have low NAM have the levels of p53 as I explained, and the entirety is Bdmv2, and that you can rescue the behavior of these things by putting NAM back in these tumors. So basically there are two pathways which in a tumor are regulated by NAM. Actually, normal cells are regulated by NAM. One is a pathway that inhibits notch, one is a pathway that stabilizes p53. When you miss NAM, you activate notch and you inhibit p53. Now, potentially, this is very dangerous for a cell, because notch is an oncogene and p53 is a tumor suppressor. So one single alteration, the lack of NAM, can activate an oncogene and inhibit a tumor suppressor. Okay, so how do we try to put all of this together? These are a few things that we know, that I told you up to this point on NAM and p53. We'll go through them rapidly. We've analyzed the stem cell compartment, and we have seen that the homeostasis of the stem cell compartment is a matter of self-fate determination. We've seen that cancer is also a problem of cell-fate determination, because the stem cell compartment is altered in cancer by altering the fate determination. But NAMB is a cell-fate determinant, and is actually involved in the decision to skew a cell towards asymmetric or symmetric division. We've seen that in Drosophila. NAMB is frequently lost in cancer. And NAMB-deficient breast tumors have worse prognosis, higher grade, and higher stem cell content. And NAMB controls p53, and alterations of p53 are relevant in cancer. So this led us to formulate what we call an ambocentric hypothesis of the regulation of the stem cell compartment in breast. It's very simple, doesn't pretend to account for all the biology, of course, but possibly for a good part of the biology of the asymmetric division in this compartment. So we believe that when the stem cell divide, one of the two daughters, which inherits NAMB, will remain stem, while the other daughter that does not inherit NAMB, because NAMB divides asymmetrically. This is an hypothesis. I will have to show you later the proof this, of course. This imposes quite different behavior to p53. Because in the cell with the high NAMB, of course you will have high p53, because NAMB stabilizes p53. I told you, high NAMB, high p53. In the cell with low NAMB, you will have low p53. Low NAMB, low p53. But what does p53 do? It blocks proliferation. So when you have high p53, likely you can have quiescence. When you have low p53, you might have proliferation. And so the progenitor will proliferate and the stem cell will remain quiescent. This is what might happen in the condition of asymmetry, in the normal compartment. In the cancer stem compartment, in the case of NAMB-deficient tumors, so not of all the tumors, but of the one that we're interested in, low NAMB, low p53, low NAMB, low p53, proliferation in both cases. Okay? So, for this to be true, a number of conditions have to be fulfilled. First of all, one should show that p53 is really involved in asymmetric divisions. We know that NAMB is involved in asymmetric division, but what about p53? Then one should show not only that p53 is involved, but that p53 is under the control of NAMB in the determination of the asymmetric division. And then one has to demonstrate that by altering this axis, NAMB p53, as it happens in tumors, you move the cell from an asymmetric to a symmetric pattern. And then you can ask a last question, which is not obligatory to prove the model, but it would be interesting, is that can you use this thing for something useful, like stem cell specific therapy? How do we approach this? There is another little piece of technology that I have to give you. Now, I've shown to you how you can isolate pure stem cells, PKH positive cells. When you put them under a microscope, you see they're red. Now you can follow the way they divide. And you will observe two patterns. In one pattern, you see that the cell divides the first time, but then one of the daughter does not divide anymore, while the other daughter divides once and then twice. And you see, of course, that the PKH gets diluted. So this is what you expect to see in an asymmetric pattern of division. After the first division, one of the two cells stays stem. You cannot distinguish it, but you can understand which one was the stem cell kind of retrospectively after you see how it behaves over time. So whenever you double in a little bit of a strange way by counting 1, 2, 3, 5, you have an asymmetric division. Now, of course, you can double in a more canonical way by going 1, 2, 4, 8 and diluting the dye progressively. And this is what we call the symmetric division. So in addition to the mammosphere test, which is a measure of symmetry versus asymmetry, but is not a direct measure, we can now also use this test, and I will show you the result also with the test, to actually count how many cells under different conditions undergo symmetric or asymmetric condition. So the first question you remember, is P53 involved? Well, did a simple experiment, well, not that simple, but retrospectively looks simple, together with Giuseppe Pellicci a few years ago. And so the idea is very simple. If P53 is involved in symmetric versus asymmetric division, you take out P53 and cells should divide asymmetrically. If they divide asymmetrically, they should become immortal in a mammosphere assay. And when you do the cell counting, symmetry versus asymmetry, they should be mostly symmetric. And this is indeed what happens. For mice, wild-type mice, P53 mice, self-limiting behavior, immortal behavior in a mammosphere assay. Do the counting on the red cells. I show you. Wild-type, 91% of the divisions are asymmetric, as predicted. P53, 84% of the divisions are symmetric. So the answer to the first question is yes, P53 is involved. But is it controlled by NAM? Is it in the same pathway as of NAM? Well, in order to do that, there are a few things that one should do. First of all, NAM divides asymmetrically in Drosophila, but we don't know whether it divides asymmetrically in the stem cell compartment. If you have pure stem cell, now you can do the experiment pretty easily. So you take a stem cell, you allow it to divide, and when it's dividing, and you can block the division also in several ways, you stain it for NAM, you know that NAM only goes into one of the two daughters. So this is good. Human beings behave like flies. In the mammary stem cell compartment, NAM only goes into one of the two daughters. But now the question is, in which daughter? Because for the NAMbocentric hypothesis, NAM has to go into the stem cell, because that is where you need high levels of P53 to block proliferation. Is this true? Yes, it's true, and you can do it like that. Can we switch the light off just for a couple of slides? So, thanks. So, this is the same kind of experiment that I showed you before. You take a red cell here, sorry, in this case it's green because we use a version of the PKH which is green, and you let it duplicate. So you see immediately that this one is the stem cell. Does not duplicate while the other cells keep on duplicating. So you can track the stem cell. And you can track it also by the PKH because this is the cell which retains the green color. Now, if at this stage you have transfected a NAM conjugated to a fluorochrome, so a NAM that you can follow in these cells, now you can ask the question, where does NAM go? And you see that NAM accumulates in the cell that accumulates the dye which correspond to the stem cell. So at the first asymmetric division, NAM goes into the cell that retains the stem cell fate. Good. But now, what does the hypothesis contemplate? It contemplates that the stem cell having high NAM should have high P53. Can we prove it? Well, we can do the same experiment by now tracking P53. P53 is a difficult protein to track, but there are indirect ways in which you can do it. Like you can take a promoter, which is responsive to P53, and hook it up to GFP. So when you transfect this promoter in a cell, if the cell has high P53 and high P53 activity, it will make a lot of GFP. If it is low, it will not make GFP. And you can do the same thing. So now the PKH comes in red, and the GFP, which means P53, comes in green. You see the cell divides. Again, it's an asymmetric division. You see that this cell does not divide while the other one divides. You follow the PKH. The red, you see, stays associated mostly with one cell. What about the green? Only, or by and large, in the cell which is also red. So in the stem cell, where NAM is present, there are high levels of P53. How does this happen? Can we get some genetic proof that really NAM is the molecule which is stabilizing p53. Yeah, we can do it by observing exactly what happens between the first and the second division. This is the stem cell, the PKH positive. GFP here means p53, as I told you. At the division, you see that p53 is divided equally between the two daughter cells. But then you see that in one of the daughter, p53 increases, while in this other daughter, p53 stays equal. You see it? Is this the stem cell or the progenitor? Well, you only have to wait for the second division. This is the cell that divides. So the one in which p53 does not increase is the progenitor. So this is good, because it shows us that the difference in p53 is not due to the fact that p53 goes differently in the two cells, but actually goes equally. But then in the stem cell, there is something that stabilizes it. So it's consistent with the NAM hypothesis. And so the idea now is, if we take NAM out from these cells, what does it happen? Well, what happens is that now GFP is a lot lower than here, as expected, no NAM, low p53 levels, is divided equally and does not reaccumulate as the cells divide, in this case, symmetrically. And you see, this is what happened in the stem cell, this is what happened in the progenitor, this is what happened in these cells in the absence of NAM. So this is a molecular genetic proof that tells you that really NAM is stabilizing p53 in the stem cell and not in the progenitor.
Can I ask you something?
Yeah.
Is a concern that GFP, the degradation of GFP might be different from the degradation of p53 inside?
Possibly. What we use here is a very fast turn, it's a p53 with very fast turnaround. It has GFP with an half-life of 60 minutes or something like that, to avoid this kind of problem. So what you measure is actually in every frame, every hour, a complete turnaround of the protein. So at the genetic level now, this must mean one thing. It must mean that NAM knockout must phenocopy the p53 knockout. And this we've done by receiving mice actually that were knocked down for NAM and specifically knocking out NAM in the mammary gland. And you see that actually the NAM KO phenocopies the p53 KO. Most divisions are asymmetric, both in the NAM KO, and in the p53 KO. And one last thing. If this is true, well, this means that NAM is upstream, p53 is downstream. If I remove NAM or if I remove p53, the effect is the same, because one stabilizes the other. So, is there a way to take a NAM negative animal, a KO, in which p53 levels are low, and the divisions are symmetric, as I show you, and somehow artificially restore the p53 level, so that to bypass the circuit downstream? Well, you can do that with a drug which is called Natalin. Natalin is a drug that has been developed to inhibit MDM2. So, when Natalin is present, MDM2 cannot ubiquitinate p53. Therefore, p53 is not degraded. Therefore, levels of p53 are increased. I'm not showing you all the details of this, but you can believe me that all the controls have been done here. But the result is actually exactly what you expect. In a NAM knockout, you have an immortal behavior of the stem cell. As soon as you hit them with Natalin, so you are doing nothing to NAM, you are acting downstream of NAM, the cells start becoming mortal and self-extinguishing. And again, most divisions are symmetric. In a NAM knockout, you treat them with Natalin, and you restore asymmetric divisions. I'm almost finished. So the answer to the second point is yes. It's actually the NAM p53 axis that controls decision in the stem cell compartment in terms of asymmetry versus symmetry. What about human tumors? We have done all of this to come, to arrive to human tumors. Well, so this is an experiment that we have done on a number of human tumors. I'm showing you 4 of them as an example. So let's say we take two NAM negative tumors. You see that there is no NAM here, and two NAM positive tumors. By immunohistochemistry, you see that these tumors are NAM deficient. Now, in accordance to everything that I showed you, my prediction here should be, these tumors are all p53 wild type, by the way, because otherwise the thing would not work. The prediction is that these tumors should divide mostly symmetrically, but if I put back NAM, I should bring them back to a normal-like state, so make them divide asymmetrically. And if I treat them with Nutlin, so if I restore the p53 level, I should also bring them back to a normal-like state, exactly like I've showed you in the mouse. And these are the controls. These tumors, of course, will also divide asymmetrically. At the end of the day, all tumors divide asymmetrically, more or less. But the alteration here is not NAM. I have no idea what the alteration is. But they are my negative control. This means that in these tumors, if I put back NAM in, nothing should happen, because the alteration is not dependent on NAM. They might or might not respond to Nutlin. It depends on whether the alteration impinges on p53. Better if they don't respond, so that the experiment is clear. Okay? But one can live also with some kind of response. So we selected tumors which are very comparable among themselves in terms of kinetic behavior. You see that in all cases, the stem cell compartment expands very rapidly in the mammosphere assay, more or less with the same kinetic. So this was done in order to have tumors that were really comparable among themselves. And so this is what happens. As predicted, these now are the symmetric versus asymmetric counting results. As predicted, blue means symmetric. Most of the division are symmetric in these tumors, most of the stem cell division. We put NAM in these tumors, so we re-express NAM in these tumors in accordance to the hypothesis. Only the tumors which were initially negative, low for NAM, recover a pattern of asymmetric division. Red means symmetric. These tumors, of course, the tumors don't care because the alteration of NAM is not relevant to their biology. What about Nutlin? We treat them with Nutlin and we phenocopy exactly the NAM restoration phenotype. So we treat them with Nutlin, and again, by recovering p53 level, you bypass the alteration with NAM, and fortunately enough, these tumors must depend on mechanism different from the NAM p53 axis, because here we treat them with Nutlin and nothing happens. And the same happens if, rather than counting symmetry versus asymmetry, we do the mammosphere passage assay, which I showed you before. The NAM-positive tumors will start to flatten towards the normal phenotype in the presence of Nutlin. The NAM-high, the NAM-positive tumors will not get. And so number 3, yes, the same mechanism that I showed you in the model system are true also in a real setting. Let's see whether we can use this to do some cancer-oriented therapy. So where is my last series of experiments? Hold for 2 more slides and we'll be done. So we take NAM-negative tumors here. We implant them in the mouse. We let them grow until a certain size. This is very important because when we do therapy only when the tumor is present. So it's very important to have a certain size of tumors before starting to do something in the eye. Then, in some cases, we treat these tumors with Nutlin. It's a brief cycle of treatment with Nutlin. I think it's 4 injections over 12 days or something like that. Then for the rest of the experiment, and the rest of the experiment will go on for months, these mice do not see Nutlin anymore. This is the only time that they see Nutlin in the entire course of the experiment. And then we look at what happens. Well, what happens is that the mice don't care. The tumors grow exactly at the same rate in the plus Nutlin and minus Nutlin. So there are 2 ways of interpreting this experiment. You throw the experiment away because you say the drug doesn't work. Or you remember the beginning of the lecture in which we said, well, if we really have a stem cell specific drug, the effect of the drug would be visible only, a long time after the initial administration of the drug. Because at the beginning, the tumor is made mostly of progenitors. If the progenitors are not effective, the tumor will keep on growing. But if you are hitting the stem cell, it's only after all the progenitors which are present at this stage will have exhausted their proliferative capability that the treated tumors will find itself with fewer stem cells and therefore with less capability of regenerating. Well, how do you do this? How do you unmask this? At this point, you have to sacrifice the animals, of course. The tumor burden is too high. You cannot let it grow. But you can transplant the tumor. So you can artificially elongate the lifespan of the tumor going into another animal. And now this is what you see in the cell line. So remember that from here to here, I don't know how long, because I'm not doing the experiment myself. Salvatore pretends that he does the experiment, but he doesn't do the meter. But there might be perhaps a month or so here of growth, and at least another month of growth with you go like that. It's 2 months. So these animals have been for 2 months without signatment. So this is all the effect that is from the initial treatment. And so you can ask, well, fine, these tumors are growing less, but is this really because they are depleted of stem cells? Well, there is a way of doing this experiment. I already mentioned to you at the beginning, you can do limiting dilution. If you do limiting dilution, then you reach the point in which the number of cells that you injected is so low that only stem cell will give rise to the tumor. And so if you inject enough animals, now you can have a frequency of cancer stem cell or tumor initiating cells. And this is what you have. This is just a sample. We have done it now on 6 different tumors, 4 non-negative and 2 non-positive. You see there is a tenfold difference in the amount of stem cell when you treat them with it. And the effect is only unmasked, in the long-term growth of the tumor. And so the answer to the last question is, yes, possibly there is at least a proof of principle way through which you can achieve stem cell specific therapy in breast cancer. And so this is my take-home message after this long lecture. The homeostasis of the stem cell compartment, at least in breast, and at least for what concerns the symmetric versus asymmetric division, is regulated by an axis in which NAM regulates p53 and stabilizes p53. This leads to asymmetric division and to a normal state. This axis can be subverted by a linked series of degradation occurrences. The degradation of NAM by a mechanism which is still not completely clear to us imposes the degradation of p53 through MDN2 because NAM does not inhibit MDN2 anymore. NAM and p53 gets degraded, and this corresponds to a situation of symmetry in the division of stem cell, which is associated with cancer. But by restoring this mechanism and by restoring p53 levels, you can achieve stem cell specific therapy, at least in a xenotransplant setting, which is the one that we used. And so these are the people that did most of the work. This is essentially all the work of Salvatore and Daniele in the lab. Ivan is the guy that initially discovered the interaction between NAM and p53. Of course, we interact a lot with pathologists and surgeons, and this is something indispensable for the work that we do. This is the Pesce stem cell ensemble. He's the one in charge of making people work together, so he makes them play together. That's the best way of having people playing together. And I think I'll stop here. Thank you.